Hi all! So I'm back and we're going to now do our second lecture, which is about the biological macromolecule proteins, or if you like, the stuff that does stuff. Okay, so this lecture talks about what proteins are made of and how they're put together, how they're shaped, and what some of their functions are, including an activity called enzymology and what enzymology and enzymes target, which are called substrates. Okay, so amino acids can be thought of as the nucleotides of protein. Proteins start as big long chains of individual molecules, just like DNA and RNA. A major difference being that DNA and RNA are once again made of chains of nucleotides. Amino acids are made of, proteins are made of chains of amino acids. So whereas DNA and RNA have nucleotides, protein has amino acids. A big, huge difference between amino acids and nucleotides, well, there are many, but here's a big one. We learned that either DNA or RNA molecule only has a possibility of 4 different nucleotides. There are 20 different amino acids that living things can incorporate. And they have 3 common pieces to them. Everybody's got a big central carbon in the middle. Everybody's got this NH2 group on one side, this is called the amine group, and everybody's got this carbon double bonded to oxygen single bonded to a hydroxyl group. This is called a carboxyl group, this whole thing together. The third thing that everybody's got, first thing, second thing, third thing, the fourth thing that everybody's got, sorry about that, is they've all got a side chain. The side chain is just kind of generally referred to as R. What makes these 20 amino acids different from one another is always contained right here in this R group. Parts 1, 2, and 3, the central carbon, the amine group, and the carboxyl group, every amino acid has this. It's what defines them as an amino acid. This is what makes them different from one another. So there are 3 different groups of amino acids, and as we can see from this, from these diagrams, here's our central carbon, here's our amine group, here's our carboxyl group. Every one of these has that same bit at the top. The parts that are different are these R's down here. They're shaded in green here, pink here, and blue or red down here. Our 3 groups of amino acids has to do with the chemistry of these R groups. In this case, all of these guys up here, R is nonpolar, meaning it doesn't interact with water, it doesn't interact with charged things. In this middle group here, where you have shading in pink, all these different R groups, the R group is polar. It's not charged. The molecule as itself, in and of itself, is not charged, but it has polarity. It will interact with charged residues, it will interact with water and other polar compounds. It does not interact very well with these guys up here. The third group is where R is charged. And we have 2 subgroups here. In some cases, R is positively charged. In some cases, R is negatively charged. And so if you put that in an acid-base perspective, these guys here that with positive charges on them are acidic amino acids. These guys here are basic amino acids. So these guys will interact with these guys up here, not particularly well with these guys down here, these guys way at the top. So how are proteins made? We know we have chains of amino acids, we already described that, but how are those chains formed? And incidentally, each one of those amino acids is called a monomer. And when you have a big long chain of them put together, that's called a polymer. Those are basic chemistry terms. A chain of anything put together like this is made of a chain of monomers, and the entire thing together is called a polymer. we'll see that the next lecture when we talk about carbohydrates as well. So amino acid polymers are the very most basic, chemically dry, boring way of describing what starts off and eventually will become a fully functional protein. So these are made of similar or identical building blocks, and they're linked together by covalent bonds. So this is, again, for anybody who needs a refresher, this is when two elements share electrons. So how does the polymerization happen? A polymerization reaction is where you have several monomers that are being joined together and becoming a polymer. They happen by something called a condensation reaction. Anytime you're doing a condensation reaction, you can also refer to this as a dehydration reaction, you have something over here that are going to be bonded together, and when they do, this is going to be left linked to this, and the byproduct of it is going to be one water molecule. So what we can guess from this, the fact that amino acid polymers are what make up proteins, is that every time we have an amino acid linking to another one of these is going to donate a hydrogen, the other is going to donate a hydroxyl group, and we're going to have a byproduct of a water molecule spit out. So to just look at this in more detailed terms, specific to amino acids, we're going to have one carboxyl group over here, and it's going to interact with an amine group of the next amino acid. These guys are going to join together. They're going to form something called a peptide bond. Now we have this monomer and this monomer linked together into a dimer, and they've spit out a water molecule. If you want to see this in a little more detail, what's actually happening is this double bond to oxygen on the carboxyl group is going to flip up like this. You can see it over here. This one of these hydrogens on the amine group is going to flip up here. You can see it here. And then these two hydrogens and this free oxygen are what are going to join together, spit the water molecule out, and then this bond is going to come over here and become the peptide linkage. If you want to actually see an animation of this happening, again, open the PowerPoint file on Blackboard and hit this link. There's a very nice animation. There's the URL for it right there, for an amino acid polymerization being done by computer animation. If you want to look at this in terms of not just a dimer being formed, but an actual peptide chain that ultimately will become a protein, here's what it would look like. You have a central carbon over here. You have, this used to be a free amine group, this used to be a free carboxyl group, and you can see that these are now linked together by peptide bonds. What you can also see in this diagram is that we've added in the R groups. So this one, you have this very inert nonpolar hydrogen, this very inert nonpolar methyl group. Here you start to have some polar groups being added. This one here, you have a couple more nonpolars. Here you have an acidically charged. So you can see not only do you have this nice chain, you have these side groups coming off with their very distinct chemistry from one another. And as we mentioned a few slides back, these ones will be able to interact with each other. These green ones will be able to interact with each other, but this green and this pink not going to interact with each other. So just want to take one moment and clarify something. What is a peptide versus what is a protein? A peptide is any small piece of two amino acids linked together, at least two. A polypeptide is a big, long chain of individual amino acid residues. A protein is a mature, structured, modified, fully folded, and ready to perform its function molecule. So as a protein is being made, it starts off as a peptide, a very short stretch of amino acids linked together. In the end, it becomes a polypeptide, a big long chain, and then after the fact, this is modified, it's folded up, and it's ready to perform its function. Now it is called a protein. Saying of which, protein structure happens on 4 different levels: primary, secondary, tertiary, and quaternary. And we'll go through each of these individually, so don't need to go through them now. We're going to start with primary protein structure. This has to do with the chemical diversity of the R groups in each amino acid. Literally, primary protein structure is defined as what individual amino acids are making up a big long polypeptide chain. So structure on the primary level simply refers to the composition of that big long chain, and that's it. What we can see by looking at single amino acid changes and the different chemistry that goes in is that a single amino acid ultimately can change the function of a protein. And there are 2 really great examples of that in human health. One is something called sickle cell anemia. So normally, the sequence of a protein that we all have called hemoglobin, which is responsible for grabbing onto oxygen. You find it in red blood cells, and bringing oxygen to all tissues in the body, is threonine, proline, glutamic acid, glutamic acid. That's what it's supposed to be. When it looks like this, we get a nice, properly folded hemoglobin molecule that is capable of carrying oxygen. And since there's so very much hemoglobin in red blood cells, it actually, the shape of a hemoglobin protein has an effect on the shape of a red blood cell. In patients with something called sickle cell anemia, what happens is one of these amino acids is different in the hemoglobin molecule. We have threonine, proline. Instead of glutamic acid here, we have something called valine here. Different amino acid. Glutamic acid is a pretty big amino acid, and it's got a charged R group, so it's very reactive. Valine is pretty small, and it's got a nonpolar R group, so it's not very reactive. This chemistry is so different that it actually horses up the entire shape of the hemoglobin protein, such that it is so different it actually changes the shape of the red blood cells. More importantly, when the hemoglobin is shaped wrong, it can't grab onto oxygen very well anymore, and it is not very efficient at delivering it to all tissues in the body at that point. Obviously, that's bad. Another great example is in a disease called cystic fibrosis. So we have, in this case, an amino acid change in a protein that's responsible for pumping sodium across cell membranes. We'll talk more about this type of protein when we talk about lipids and membranes. We'll get back to that at some point. But the idea here is that if you do not have this protein shaped properly, it does not pump out sodium from in and out of a cell properly, and the actual functional result of that is that you have, instead of nice clear mucus, you wind up with very sticky, osmotically wrong mucus. If you are a healthy person with the right protein sequence, your lungs normally look like this. There shouldn't be a lot of sticky, gooey, yucky mucus in them. If you have cystic fibrosis, it's not that you have an amino acid swap in your gene sequence, it's that you're actually missing this amino acid right here called phenylalanine. So instead of having asparagine, isoleucine, phenylalanine, glycine, valine, you just have asparagine, isoleucine, nothing, glycine, valine. This is a real big amino acid. It's nonpolar, so it doesn't have a lot of reactions associated with it, but it's got a really, really large shape to it. And when this is suddenly gone, it very much changes the shape of this protein, which again is this one up here, responsible for regulating the amount of salt going in and out of the cell. This is an x-ray from a cystic fibrosis patient, and you can see there's all kinds of white, gooey, cloudy looking stuff in here, and this is actually an image of this thick, sticky, gooey mucus. point being, one change in this primary amino acid chain, primary protein sequence, is going to greatly, greatly alter the overall structure and chemistry of the protein that it's meant to encode, and it can greatly change the overall function of a single cell, a single tissue, and ultimately a single human being. On the other hand, secondary protein structures, where we're going to add another level here. We're going to start looking at the interactions between these R groups, not just what chemically is there. So secondary structure is technically defined as the bending of this polypeptide chain and the hydrogen bonding that results from that. So some of the most common shapes that we can make within a single polypeptide chain are an alpha helix or a beta pleated sheet. This is a diagram of an alpha helix. This is a diagram of a beta pleated sheet. What's going on here, I'll show you a better picture in a second, is that some of these amino acids that are not next to each other in the chain are interacting with one another via hydrogen bonding between their R groups. So this is a better diagram of that. In an alpha helix, you might have every fourth, it looks like in this case, residue hydrogen bonding to one another. Depending on the periodicity of this, you'll get a nice tightly coiled helix or a fairly loose one. You have to have the right R groups that are going to be able to interact in an evenly spaced way in order to form this shape. On the other hand, you can form a different shape, this beta pleated sheet. In this case, we have this amino acid here and this one here interacting with this one here next to this one here. So you can see these interacting residues are not evenly spaced the way these are. They're one next to another with a whole bunch in between, same number in between, and then one next to another again. So you get these kinks, and you get this big fold, and you get kinked lines lining up next to each other. An important point that's made in this figure right here is that the same polypeptide chain can have both of these shapes numerous times. So this looks like one end of the chain here. We've got a beta sheet forming here between these sets of amino acids. If we follow the chain a little further, right here it makes an alpha helix. Up here it's folding again, it's not forming any structure at all, this white tube right here, and then now it's going back and forming another beta sheet. So again, it's not that a chain forms either a beta sheet or an alpha helix, it can actually form both at different points in the chain. Tertiary structure. We now have an amino acid chain, it's curled up into, in this case, alpha helices. What goes on in tertiary structure is that you start to have hydrogen bonding between either these empty spaces that don't have secondary structure, or even between secondary structures themselves. So this is giving another layer of shape to what used to be a flat, straight polypeptide chain. Quaternary protein structures, the last level. It's very important to point out that not all proteins have quaternary structure. For the ones that do, what this means is that some proteins have several subunits that come together and form one active protein. This is a diagram of hemoglobin, which we were talking about a second ago. Hemoglobin as a large protein is something called a homotetramer. Homo means same, tetra means 4. So each one of these different colored blocks here represents an independent, separate polypeptide chain. We need these guys to be made as a single straight chain, fold up into many alpha helices, and then 4 of these coiled up subunits are going to come together and interact with one another to make one big, giant, functional hemoglobin protein. So there's 4 pieces to this that all have to be there in order for this hemoglobin protein to function. Each one, again, started off as its own separate chain. I've got another example right next to this called reverse transcriptase. This is a protein that's made by an HIV molecule, HIV virus. The reason I have this in addition to this is that reverse transcriptase is a type of protein called a heterodimer. Hetero means different, dimer means 2 subunits. So reverse transcriptase has 2 different subunits, 2 different pieces that have to come together, and they're not the same. So over here, hemoglobin had the same 4 polypeptides to start with. Reverse transcriptase has 2 different polypeptides that are going to fold up slightly differently and interact with each other slightly differently to make one complete reverse transcriptase protein. So this is an image, and you can pause this and take notes on it if you like, of the different levels of protein structure. Okay, so the last thing we want to talk about here are enzymes and substrates. Enzymes are proteins. If somebody talks about an enzyme, they are talking about a protein. If they are talking about a protein, however, they are not necessarily talking about an enzyme. There are types of proteins other than enzymes. Enzymes are just extremely biologically active proteins, and therefore they tend to mediate a lot of different functions. The word substrate is linked with the word enzyme because the enzyme is the functional component of this relationship. The substrate is the thing that gets acted on. So substrates are specific substances that are targeted by an enzyme. So enzymes are usually very, very specific, meaning that one particular enzyme is going to target one type of substrate, and it's not, by and large, going to interact with anything else. But that's not always the case. For example, there are certain types of enzymes called nucleases that chew up nucleic acids. There are some made by protozoans. We'll talk about that later in the semester. And they just blindly chew up all nucleic acids. It doesn't matter what the sequence is. So enzymes and catalysis. Catalysis is a process by which a certain activity is initiated or made energetically favorable. This is why you typically hear the phrase enzymes are catalysts, or biological catalysts. I heard that a lot in high school. I never really had a good appreciation of what it actually meant. If you think about what the word catalyst means from a linguistic perspective, it's simply something that kicks off a process. So enzymes are good at kicking off biological processes by starting reactions in one of 2 ways. They either bring 2 molecules together that normally wouldn't be together and allows them to interact, or it lowers their activation energy. It makes it energetically favorable for those 2 things, those 2 components to react with each other. By doing either of those things, it speeds up the process by which a reaction between 2 biological molecules in a cell is going to actually happen. So I have 2 videos here. One of them is goofy, one of them is not. This is actually a computer animation of an enzyme and a substrate interacting with one another. It's very good if I sound like I'm talking nonsense right now. This one is good for remembering. This is actually a student-made video for enzymes and substrates and the point of catalysis, and it's funny, but it has some salty language in it, so you've been warned if easily offended. Okay, so regulations of enzymes. Why would an enzyme be regulated? Well, for starters, this is an energy issue. We don't need all cellular functions to be happening at any given time. That's typically a waste of energy. We don't need all of our enzymes that are responsible for metabolizing the sugar lactose to be turned on if we haven't got any lactose being fed to us. It's a waste of energy to have them kicking around. So enzymes are typically regulated. They're regulated on two different levels. They can be regulated on a genetic level, meaning the gene that encodes that enzyme can either be turned on or turned off as appropriate. In addition, they can be regulated on an inhibition level. You can do something called competitive inhibition or allosteric inhibition. Competitive inhibition is where you have a fully formed enzyme and you have something called a blocker or some kind of regulatory molecule that's going to sit in the site where normally its substrate would sit. So if this is the substrate that your enzyme is acting on, and you've got these little properly shaped grooves here, and for whatever reason we don't want this reaction to happen, we can have this regulatory molecule that's going to sit in this shape and outcompete the substrate. These binding sites aren't free to interact with the appropriate substrate. On the other hand, we have something called allosteric inhibition that can happen. This is where we have another groove on the enzyme that is completely separate from where the substrate is supposed to sit. But if this regulatory molecule is put into place, these grooves are going to change shape and these substrates won't be able to bind. If we then want that enzyme to be active, we can pull a regulatory molecule off. It will resume a shape that the substrate's able to bind into, and then the reaction will be able to happen. This is a little bit hard to grasp, so I would strongly suggest watching this animation. Again, there's its link right there, or you can download the PowerPoint file and play this, but I would strongly suggest watching this animation of that. It's very helpful. Okay, so the last point, or no, second to last point I want to make here are enzyme names. Not every single enzyme, but the vast majority of them end in the suffix ase. So if you see a word that ends in ase in biology, you know it is an enzyme. So anything ACE, if you like, would be an enzyme. The other cool thing about this is that the prefix or the root of the word in many cases describes the function of the enzyme. So whatever this root word is, it's usually refers to an enzyme that either adds, degrades, targets, or performs the function of whatever the root is. So I have a few examples. First of all, the enzyme sialidase cleaves sialic acid. The enzyme phosphorylase adds phosphate groups. The enzyme glycyl synthase synthesizes glycine. So in this case, it degrades the root. In this case, it adds the root. In this case, it does the function. My point here is that you can define an enzyme that you've never ever seen before simply by looking at the word. So I can give you a big long giant enzyme name, and as long as it ends in ase, you should be able to know that that is an enzyme and it targets the big long thing at the front of that word. This was particularly helpful if you are a pre-med major and you would like to take the MCATs someday. Having a strategy to have a pretty reasonable guess at what the function of that word you've never seen before in your life does is extremely helpful. Okay, so the last point I want to make, and this is the last time I'll bring this up about the first living biological entities. In the previous file we were talking about nucleic acids, so I've already kind of blown the answer to this, but the question was the first living entity a protein? So arguments in favor of this, in Stanley Miller's prebiotic soup that he made, amino acids were very abundant macromolecules, so they very well could have been existing on the early Earth. They're very efficient at doing things. They're very efficient catalysts, as we say. But what we need for this first entity is a self-replicating molecule that has the ability to act as a catalyst for the assembly and polymerization of its own copy. Therefore, the argument against this is that a protein would need to have a template in order to replicate itself, and it would have to have the ability to store information. It does not have either of those things. It would be perfectly able to catalyze a reaction replicating itself, but it does not have a template, and it cannot store information. So, probably not a protein. And that is the last thing I have to say about proteins. So, I will see you next time.